Welcome to the third series of lectures in this course dedicated to exploring the world of instruments and transducers, mainly focusing on their application in medical settings. Throughout this part of the course, we'll delve into some fundamental principles, technologies, and practical implementations of these vital components in healthcare. Let's begin. In this part of the course, we encounter a wide variety of technologies and measurement techniques. However, our approach will be highly selective. Instead of attempting to cover a broad range of techniques superficially, we're going to focus on a few key methods. This selective approach aims to foster a deeper understanding of these important techniques, allowing for more comprehensive learning and mastery. Given the breadth of material available, it's important to note that while we will not cover every technique in detail during lectures, there are optional reading assignments that can provide valuable content and additional information. Selectively engaging with these materials can help reinforce and expand your understanding of core concepts. Additionally, closely following the lecture slides and reviewing them after class will provide a structured framework that highlights the most essential techniques and concepts. This combination of focused study and selective reading will equip you with both a strong foundation and a broader perspective on the subject matter. The primary focus at the start of the last four series of lectures in this course will be on understanding ion-selective electrodes and their clinical relevance, setting the foundation for more advanced concepts in later lectures. Module 11 on ion-selective electrodes. Basically, this module provides a comprehensive exploration of ion-selective electrodes and their clinical applications. First, we have an overview of ion-selective electrodes, their working principles, and their importance in analytic chemistry and clinical diagnostics. Then we'll move on to discussing the role of the electrolytes in maintaining physiologic balance, highlighting their clinical significance, and then the necessity of accurate measurement. The third lecture of this particular segment is a deeper dive into the design, functionality, and applications of ion-selective electrodes, ISEs, emphasizing their use in measuring specific ions in complex solutions. The fourth lecture in this series for Module 11 is on amphiometric transducers. Exploration of amphiometric transducers and their application in electrochemical sensing, particularly in the contents of biomedical sensors and measurements. We then move on later to modules 12, 13, and 14, and we'll highlight those in subsequent lectures. In the modern landscape of medical diagnostics and monitoring, the accurate and timely measurement of physiological variables is paramount. From electrolytes to glucose levels, pH, and beyond, the ability to precisely assess these parameters is crucial for patient care, disease diagnostics, and treatment management. Central to this process are instruments and transducers, of course, it's subject to this course, which serve as the interface between the physical world of biologic signals and the digital realm of data analytics and the interpretation of the analysis that ensues. The devices we speak of translate biologic phenomena into measurable electrical signals, enabling healthcare professionals to obtain vital information about a patient's health status. Among the myriad of instruments and transducers, ion-selective electrodes stand out as an indispensable tool for the measurement of electrolytes, such as sodium, potassium, chloride ions, and biological fluids. These electrodes exhibit selectivity towards specific ions, allowing for accurate and selective detection even in complex biological matrices. Moreover, the measurement of glucose levels, a cornerstone in the management of diabetes and other metabolic disorders, relies heavily on advanced sensor technologies and transducers. These devices employ enzymatic reactions or electrochemical principles to quantify glucose concentrations in blood or interstitial fluid with high sensitivity and selectivity. Furthermore, pH measurement, representing the acidity or alkalinity of a solution, holds critical importance in various medical contexts, including blood gas analysis, tissue culture, and physiologic research. Transducers designed for pH measurement enable precise monitoring of physiologic pH levels, providing valuable insights into metabolic processes and pathologic conditions. In fact, the project for this part of the course will be focused on pH measurement. It's pretty straightforward, but it's important to pay attention when we get to that part of the course, and we'll have Q&A sessions to help navigate you with that project. Still, from the figure shown for Part 3 of this course, the focus is on non-electrical sensors and transducers. Electrical sensor analysis was covered at the beginning of this course. A biosignal is any signal in living beings that can be continually measured and monitored. The term biosignal is often used to refer to bioelectrical signals, but it may refer to both electrical and non-electrical. Your journey so far in the course has involved electrical biosignals involving the change in electric current produced by the sum of an electrical potential difference across a specialized tissue, organ, or cell system like the nervous system. Electrocardiography is an example that you work through in detail. In the remaining 4 modules of this course, modules 11-14, we explore non-electrical signals that are capable of being monitored from biological beings such as chemical signals, acoustic signals, and optical signals. Important word about transducer versus sensors. All sensors are transducers, but not all transducers are sensors. Simply stated, a sensor is a device that detects and responds to a specific input. Again, this was covered in earlier modules. You discussed EKG sensors measuring electrical signals produced with muscle contractions. Most often people clinically look at lead 2, but a more comprehensive system is one that I think you explored previously. In comparison, a transducer is a device that converts one form of energy to another, such as converting mechanical energy into electrical signals. So for instance, a strain gauge is a transducer whose resistance value changes in proportion to the strain it experiences. We tend to use the terms transducer and sensor interchangeably, but in principle, they're slightly different. This is just FYI. The figure presented is a flowchart illustrating the typical components of a biomedical measurement system. The primary emphasis is on biosignal sensor and transducer subsystem, which plays a critical role in detecting and converting physiologic signals into measurable electrical output. A key concept in this system is the term measurand, which refers to the specific physical parameter being measured. While the term may seem complex, it simply denotes any quantity that is quantified and compared against a predefined standard. Examples of measurands include temperature, voltage, pressure, and height. Throughout this module, the focus remains on understanding the sensor portion of the system, highlighting how these sensors capture and process biosignals before further analysis through analog and digital components. This particular slide builds on the sensor block introduced in the previous slide, providing a more detailed breakdown of the interaction between analytes and biorecognition elements within the system. The analytes, also referred to as the sample matrix, can take many forms, depending on the application. These include antibodies, used in immunoassays to detect specific antigens, enzymes to facilitate biochemical reactions and are often used in biosensors for glucose and other metabolites, microorganisms employed in microbial biosensors to detect specific pathogens or assess environmental conditions. DNA, of course, used in general assays for detecting mutations, pathogens, or specific sequences. Cells used in cell-based biosensors for toxicity assessment or even drug discovery. Electrolytes, okay. The primary focus of this module, which includes ion-selective electrodes that measure specific ions in biologic fluids. Biorecognition elements selectively interact with the analyte, initiating a response that is then captured by a transducer. The transducer converts this biologic interaction into a measurable signal, most often an electrical signal. This signal can then be processed, analyzed, and interpreted for diagnostic or analytic purposes. In this module, the focus is on the electrolytes category and their detection through ion-selective electrodes, which play a crucial role in clinical diagnostics. Again, in this module, we explore the design of electrochemical cells, which varies depending on the measurement principle being used. Of particular interest are electrochemical sensors that operate primarily on three techniques: the potentiometric measurements, involving the measurement of voltage to determine ion concentration; amperometric measurements, measuring the current generated by, say, a redox reaction to quantify analytes; and then conductivity measurements, assessing the ability of a solution to conduct electricity, often related to ion concentration. We will review this slide later in the course to review and track the techniques we've explored as we progress through the material. In essence, this module, many different doors to potentially open, and of course we could dive into many different topics, but for the current work, we're just going to look at three electrochemical measurement techniques: the potentiometric, amperometric, and conductivity measurements. But again, to be clear, most of our focus will be on the potentiometric and amperometric measurement techniques. For those new to the field, we'll again only opening a few of the many doors possible, and the next lecture talks in more detail about the clinical relevance of electrolytes, and this may be review for many class participants.